Welcome back to the Neurostimulation podcast, where we dive into the science, innovation, and clinical frontiers of neurostimulation. I'm Michael Passmore, and today we're going to be having a great conversation. I'm thrilled to be speaking with Dr. Jennifer Rodger, professor at the University of Western Australia and head of the Brain Plasticity Research Group at the Perron Institute. Dr. Rodger's lab is pioneering research into the mechanisms and safety of low-intensity repetitive transcranial magnetic stimulation, or rTMS, particularly in the developing brain. And so we're going to be discussing, among other things, a recent study that explored the effects of rTMS on adolescent mice, what it reveals about brain plasticity during critical developmental windows, and perhaps the broader implication for using neuromodulation in younger humans as well for clinical purposes and also other types of research endeavors. Dr. Rodgers, thank you so much for joining us. It's really a pleasure to meet you, and I'm really looking forward to the conversation today.
Thank you, Michael. Me as well.
Great. So maybe if you don't mind, I'm taking a few minutes just to introduce yourself to us and help the audience to understand, a bit more about your work and your experience and what your lab is focusing in on these days.
Yeah, thank you. Please call me Jenny. I'm a neuroscientist and I'm based in Western Australia in the beautiful city of Perth. And I suppose my background is a little bit unusual because I come from a biochemistry background. So I studied organic chemistry and biochemistry and molecular biology, but I always wanted to get into neuroscience. And over the years, I've studied a range of different animal models of neuroscience. So I've always had an interest in development, brain plasticity, how the brain repairs after injury. And as I developed my own research stream emerging through postdocs and various different lab experiences, rTMS was a sort of mystery to me. And I have to admit that I was a TMS skeptic when I first started working in this field. And I had a number of people around me who were saying, it's amazing, it changes the brain, we can use it to treat all of these things. And I was very skeptical. And when a colleague challenged me and said, well, what would it take for you to believe in TMS, to really believe that it works? I said, well, no, we should look in animal models because we can actually then get inside the brain and look at pathways, look at cells and see the effects of TMS on the brain. And that would convince me if we could see things. And so we applied for a grant and we got the grant. And that's where my research focus now is based around TMS, understanding how it works, how it can be applied. And that all stems from that initial argument and grant application to convince myself that rTMS was actually having an effect on the brain. So now my research extends not only in the mechanisms, but we started working in translational neuroscience, trying to find ways to translate basic findings into human practice. It's challenging. I can't say we've had any major successes yet, but it's been a really fascinating journey. And I look forward to talking about lots of different aspects of this with you today.
Yeah, fantastic. That's so interesting. Thanks so much for explaining that. I think I've talked to so many people who, myself included, who have been skeptical about neurostimulation. And I think part of what's interesting is because I'm thinking of this, I'll put it up, I'll show this picture that I'm thinking of, but it has to do with the history of magnetism and how 100 years ago people were doing things, putting magnets towards the head. And so it does engender a sense of skepticism, and it lends people understandably to wonder to what extent this is a scam or there's snake oil kinds of clinical applications. I think even currently there's this potential for unscrupulous people to be producing devices that are just nothing more than shams. But now 20 years on, after really it's been starting to kind of catch on clinically, there's so much evidence. And I think understanding more about the basic research that you and your team are involved with is extremely interesting. Yeah. So I'm looking forward to talking about it also.
Thank you. Yeah, I think it's also, TMS is unusual, as you pointed out, but it happened and the medical device suddenly became used and employed everywhere. And unlike a drug or, most common interventions, there hasn't been that body of evidence built up before translating to humans. So it's almost working backwards. And now we've got something that works sort of most of the time, or some of the time. going backwards and trying to understand why it works and why it doesn't work in some cases. I think it's a really interesting area.
Mhm. Yeah, yeah. And just thinking about this particular study in terms of the animal model, it just brings back memories. I did some work study kinds of terms in undergrad in animal biology lab at UBC in Vancouver here. And so I can recognize the value of this kind of research. And I'm particularly curious about the logistics of developing miniature TMS coils, rodent-sized coils. So help me understand how that all worked. That must have been an interesting journey.
It was a very interesting journey. I have when I give talks, and I have a slide where I show a human head with a human TMS coil and saying, and so, I got this grant and we were funded to do TMS in mice, and I put up a mouse to scale with the human head, and everybody laughs because you see how huge the coil is relative to the mouse. And there's no way that we could use a human coil, at least in my opinion, and having spoken with a lot of other people. The strength, I think, of human TMS is that it delivers focal stimulation to a particular place in the brain so that you can target particular networks and, alter connectivity. And as we know more about it, we think that focality is really important. But when you try and do that in a mouse with a big coil, you're stimulating the whole animal. So it was really important for us to try and find a way to miniaturize the coils. And two people really were instrumental in that. A colleague, Professor Rachel Sherrod, who works in Paris, she was the one who actually said, we need to miniaturize these coils. And she developed some prototypes, and we had lots of long chats and arguments about it. But then I had an amazing PhD student, Alex Tang, who's now got his own lab in TMS and brain aging, particularly. And for his PhD, he worked with physics department at our university and wound coils on a proper coil winder and developed some modeling skills using electromagnetic field modeling programs like COMSOL. This was before things like SIMNIBs existed. So it was really hardcore physics and electromagnetism theory that we got into. And it took him the best part of a year to get a working prototype. We had to buy power supplies. We had to buy waveform generators, look at the different shapes of the pulses. And yeah, it was quite a journey, but those small coils are fantastic because they can stimulate a quarter of a mouse brain, which I think it's not as good as in humans, but it's getting there. The only disadvantage is that because the coils are small, the intensity of the magnetic field is very weak. So that means that we can't replicate motor evoked potentials that are such classic conventions in human TMS. And we can't deliver stimulation at the intensities that are clinically relevant. So that's led us to take a number of different pathways. In the study that you spoke about, looking at developing brains, we've had to focus on that concept of perifocal stimulation, because even though you have a core of high intensity stimulation at the hotspot, there's a sort of surrounding, I don't want to say aura, but lower intensity emanation of magnetic field around that hotspot, which is still activating brain cells and having an impact. So from that point of view, I think the coil can be useful in understanding some aspects of GMS, but it's clearly not a perfect model. But then no model is perfect.
Right. Yeah. Well, it's interesting just by itself in terms of what effect that type of magnetic field does cause and what the observations are and how those changes might then be relevant in terms of clinical applications.
Exactly. I hope that it's contributing. Sometimes I ask myself how relevant these models really are. But then if we can get effects with low intensity magnetic fields, then do we really need high intensity magnetic fields? Are there opportunities to change the parameters and explore other mechanisms that might have more impact in other disorders or have different effects on brain cells? Again, I think all of that discovery is important.
Yeah, for sure. And I think it makes sense just in terms of understanding the emerging breadth of potential neurostimulation applications that are coming out, whether it's TDCS or transcranial focused ultrasound, or so it's clearly not just limited to the specificity of focal high frequency or low frequency rTMS in a human model. So it's super interesting. So I've been given what you're just saying, what kinds of, I guess maybe if it's presumptuous to ask, or if we're not quite there yet, but like, what do you envision then as perhaps a potential translational bridge between what you've learned with rodent studies and potential clinical applications for humans down the road?
Well, I suppose the most advanced example of that is work that we've done with Kayleen Young, a colleague in Tasmania at the Menzies Institute. And a few years ago, we started working out what is TMS doing not only to neurons, but also to other brain cells. And the brain isn't just made up of neurons. There are astrocytes, there are oligodendrocytes, but all the blood vessels, microglia, and their colleagues in Germany who are doing a lot of work around microglia. But the work with Kayleen was interesting because we focused particularly on oligodendrocytes. Her lab is expert in multiple sclerosis research. And she found that the low intensity stimulation would improve the survival of oligodendrocytes. And in mouse models of multiple sclerosis, it will improve demyelination. It will increase the amount of myelin that they produce, that the cells produce. So it's actually a completely unexpected outcome of rTMS, electromagnetic stimulation of the brain, has the potential to improve myelination. And we've translated the circular coils in mice, stimulating large areas of the brain in humans. We've measured equivalent magnetic fields so that the cortex of the mouse was receiving a certain intensity, and we've matched that in humans through, again, a lot of modeling, back of the envelope calculations. And we're in phase 2 clinical trials now looking at efficacy of that. So for me, it's not really my story, it's Kayleen's story, but it's been a great privilege to be involved in that and see how some really basic questions that our devices help to ask have actually led to some new innovations in human trials. So I'm hoping that for things like depression and anxiety, our work will again contribute to that in the future. But I think there's a lot of potential for there to be a strong translational pipeline between the animal models and the human practice.
Mhm. Yeah, absolutely. That's fascinating. I think that's really interesting to think about how these technologies are affecting other systems in the brain aside from the neuronal systems and pathways, and connectivity and neuroplasticity. The glial cells are often just under-recognized in terms of their importance. I'm sure it's just a limitless kind of area for investigating with these kind of technologies. So the other part of it that I was fascinated by is just this idea of the impact of the TMS on the developmental piece, the neuro developmental sort of change there. So I'm just curious, if you don't mind, that particular study. So as I understand, it was targeting visual cortex during an important period of the mouse development. So can you tell us a little bit about that and what the study basically yielded in terms of findings?
Yeah, that's a study that we're particularly interested in at the moment. TMS is approved in adults for treating major depressive disorder. And one of the real attractions, I think, of TMS is that there are very few side effects. And we know that young people who experience depression and anxiety they really don't respond to medications very well. The medications are often developed in adult. They're not well adapted to use. And there's a lot of side effects. Youth themselves feel a little bit disempowered when they just get given a pill and told to go away and get better. So there's a lot of interest in trying to find new treatments, and TMS has been trialed in young people. There's reasonable, I'd say actually very good safety evidence that there are no side effects beyond what you see in adults, and the young people tolerate it well and they seem to respond reasonably well. The efficacy, I think, is still not particularly, there's not a lot of evidence yet for the efficacy. But because the developing brain, in humans, the brain continues to develop until roughly the age of 25. So if you're stimulating the brain in a young person, it's sort of an opportunity that could be fantastic because the brain is very plastic in a teenager or in a child. So you have great potential to modify it and to repair anything that might be abnormal circuitry or neurodevelopmental disorders. But you also have a big risk that any changes you make in that period have the potential to affect lifelong brain functioning. And in humans, we've had TMS in the population for probably more than 20 years. So I think there's a lot of confidence in adults that we're not predisposing people to neurodegenerative conditions, and we're not causing lesions, and we're not altering DNA or giving people cancer. So we're confident about that for adults. But children really are, I don't want to say vulnerable, but susceptible to change. And one of the questions that we've had from speaking to young people with anxiety and depression is, we love the idea of TMS, but what's my brain going to look like in 20 years or in 30 years when I'm 80? is it going to impact who I am and how my brain works later? So we thought, well, we can't do that in people because nobody's going to fund us for 50 years. Much as we would love to get funded for 50 years, it's not going to happen. So we thought animal models are a perfect way to look at this. We can take a young mouse. The adolescent period is roughly up to about 40 days. Give the animal TMS during that period and then just see what happens. And the paper that you mentioned that we published in Brain Stimulation last month really describes that short-term outcome. So we did the very simple experiment. We gave the animal our low-intensity stimulation, arguing again that we're looking, because we can't deliver the focal stimulation to the prefrontal cortex in mouse, we can still deliver the perifocal, the low intensity that a child would receive. And we stimulated the visual cortex because in mice, it's very difficult to measure things like mood and depression, and those models are quite controversial. Whereas in the visual system, you've got a great readout of the organization of the visual system connectivity, because there's a point-to-point mapping between the retina and all of the visual brain centers. So the actual connectivity is easy to measure. You then also have a behavioral readout. Can the animal still see? Can it respond to visual stimuli? So although it does abstract the model away from humans, it's actually a really powerful model because it allows us to examine the anatomy, the cellular composition, and the behavior, I suppose, in the reverse order. You look at the behavior first, and then in those same animals, you can euthanize the animals and then examine the brain structure. So that was the idea behind the study. We wanted a really clear readout of the structure and function of the brain after it had received TMS during development. So the results are compelling, and there's a lot more to do. But in a nutshell, there was no impact that we could find on any of the anatomical organization of the brain. There were no changes to any of the neurotransmitter markers, but I'll come back to that later. There didn't seem to be any inflammation. We used some fairly broad tools to look at that. But there was a behavioral change, and that really puzzled us because we didn't expect to see that. And the behavioral change was very specific. It was a visual motor integration. So the test that we use is called an optokinetic nystagmus test. And all that means is it's basically your eye tracking movements. So when you sit in a train or a car and you're looking out the windows, your eyes flick back and forth as the landscape passes by, because it's a reflex. We can't really control it that well. But our eyes follow moving objects. And you can do this in mice. You put them in a moving circular drum. And as the stripes move past the animal, their heads and eyes track the stripes, and you can count the number of times that they do that. So after TMS, we found that the mice did less tracking. They essentially didn't follow moving stripes as often as control mice did. And so we went back to the anatomy and we looked at the different regions we had traced and found absolutely no change. And though we looked at the main visual pathways, but there are always more. So we can't rule out that other visual pathways might be disorganized. But because the deficit involved both vision and movement, I think it's actually a really interesting idea to think that TMS might have compromised some of that sensory motor integration pathways. So it's not just about vision, it's about how vision integrates with movement. And in development, those processes are really complex. They're still developing. And it's possible that by providing stimulation and by providing additional activity, TMS essentially increases the electrical activity in the brain. We could have compromised development of these pathways. So is that important? Does that strike fear into my heart for TMS in young people? It honestly doesn't, partly because we're looking at a model that's very abstract. We're looking at a visual system. We're looking at sensory motor pathways. And the deficit was very small, and the deficit happened immediately after TMS. So we're thinking probably if we stop TMS and let the animals just live normally for a couple of weeks, the brain would probably restore normal function because that's what the brain does. And especially in young people where the brain remains plastic for a long time.
Sure.
So we don't really know what would happen in an abnormal brain. So a brain that had connectivity related to depression or anxiety-like symptoms. Or what is the equivalent of sensory motor integration in those brain regions involved in mood regulation and emotional processes? So I think it's interesting. It raises questions, and it certainly raises just concerns that there is something going on in the young brain that doesn't probably happen in the same way in an adult brain.
Hmm. Yeah, that's really interesting. I mean, it's certainly reassuring that there were no concerning findings on the cellular and neurotransmitter and inflammation side. Yeah.
Yeah.
I can see how there's caution that's required in terms of interpreting those behavioral findings in terms of the applicability to humans for sure.
Yeah, I think the neurotransmitter question was tricky. We did additional experiments in response to reviewer comments on the papers saying, well, if it's not anatomy, what is it? You have to have an explanation. So we went back and looked at the excitatory and inhibitory balance in the brain. But again, animal experiments are quite tricky because we wanted to use the same animals for everything. So we have behavioral outcomes in the animals, we have the anatomical tracing, and then in the same brains, we actually studied the neurotransmitter and inhibitory excitatory balance. But after TMS is stopped, by the time you've done the behavioral experiments and the anatomical tracing, it takes about a week to 10 days to do all of that. So the effects of TMS are already starting to wear off at the time that you euthanize the animals and start to look at the neurotransmitter profiles. So no, we were really limited in how much we could interpret that because so much time had gone by. So at the moment, we're continuing on from those experiments. We're hoping to do some long-term studies, not even 3 months after the TMS, but animals live for roughly 18 months to 24 months. So that is within the capacity of funding body where we can have studies that TMS mice in youth and study them all the way into old age. And that will give us a much more detailed time course of what's going on. And we have the capacity also to take different cohorts of animals to specifically address questions like excitatory and inhibitory balance, as well as putting them in the MRI machine, scanning brain connectivity and getting a much bigger picture of it.
Mm-hmm. Yeah, that's fascinating. I mean, that totally makes sense in terms of the next steps and how to sort out all of these questions for sure. And I think I certainly feel reassured as well. I'm glad to hear that you're being right immersed in this work are also reassured around the likelihood that this is going to be a safe clinical application in children and adolescents. And I guess just thinking about the recent FDA approval of rTMS in adolescents with major depressive disorder, I think that, as you said, not only the long, now 20 plus year history of effectiveness and safety in adults is reassuring, but I also think maybe this is more of a general speculation based on the idea that young brains are more plastic in general. It would strike me that then would by itself be a reassuring factor in terms of, well, if we're imposing this energetic change on the brain in a therapeutic sense, then hopefully the baseline neuroplasticity of the developing brain is almost buffer that would help to guard against any long-term side effects or other morbidity issues that might some level be conceivably related to the short-term application of the magnetic field.
Yeah, I completely agree. And I think also this is a whole other question, but we don't know if the same parameters that work in adults are also effective in humans. We have a fairly limited, the parameters in the TMS are unlimited. Any combination of frequency and intensity and number of pulses, it's a bit of a scary area to go into. There's just so much possibility. But in adults, we've sort of narrowed it down to maybe 2 or 3 frequencies that we think are effective and that seem to give good outcomes. Whether those work in children or whether it's worth actually re-exploring some of those stimulation parameters to try and find something that's more specific to children, I wouldn't even know how to start going about that, apart from attacking that huge parameter space. But I think that it's a bit like with drugs. We can't just assume that children are small adults. They have different brain chemistry, they have different brain structure. Neuroplasticity is hugely significant at that stage. So I think it's important to think carefully and make sure that we do TMS properly in young people.
Mm-hmm. Mm-hmm. Yeah, it makes sense. And I think it also makes sense in terms of what I hear a lot of people talking about, and it makes perfect sense, is just this idea of tailoring protocols to an individual's needs. So whether it's age on the young end or even on the later life end, or other aspects of particular neuropsychiatric disorder or other I'm not sure. I mean, anything just as a medication, I think probably this is a limitation of the legacy medication. I think obviously psychotherapy, there's more room for tailoring. It's necessary, obviously, but on the medication side, perhaps not, right? So maybe with a multimodal approach to treatment with tailored protocols, depending on what research shows across the age spectrum, it makes sense that this is the sort of work that would help to inform those eventual clinical guidelines and protocols being accepted.
Yeah, no, absolutely. And TMS is actually really well set up for being tailored, because there's choice not only around the region that's being targeted, but also all of that then sort of electromagnetic parameters with the pulses and the intensities. And I think we've seen with things like the SAINT protocol coming out that there is a lot of opportunity for tailoring. There's quite a lot of excitement around even just very simple tailoring brain regions based on connectivity patterns. So I think there's, we've taken the first steps, and it's now open to a lot of opportunities. And as you mentioned, I think the combinations as well, TMS combined with medication, TMS combined with psychotherapy, those sorts of really integrated treatments are really exciting. And for youth where we know they really want to be involved, well, the youth that we've spoken to in Australia, they really want to be involved in their treatment. They want to feel like they're empowered to be part of the decision-making process. And I think the opportunity to tailor treatments and to get them engaged in making decisions around that is going to be really powerful, because that will also support their own mental well-being.
Definitely. Yeah, definitely. I think that's a part of the issue historically with the legacy approaches is exactly as you say. It's the way that clinicians kind of get sucked into the whole pharmaco-industrial complex, and they're only insured for a limited amount of time, which makes it easier, shall I say, to write a prescription and book an appointment in a 2 weeks time thing. And I can see how already younger folks would be already disinclined to come in the first place. And then if that's the sort of approach, then they're justified in having a skepticism around that. So hopefully these kinds of novel treatment approaches and more ability to ask questions and be interested in the technology, that will help to foster a sense of agency and involvement with their own treatment. Definitely. It's fascinating. The other thing that's interesting that I was thinking just now is the potential for perhaps the tailoring to involve not only, as you were just saying, a given neurostimulation option plus medication plus psychotherapy, but different kinds of neurostimulation options like TMS, and then maybe something like TDCS for maintenance treatment, something along those lines. And it's also interesting to think about, I'm wondering a bit about whether you're based, it's probably too early to say, but if in terms of thoughts that you and your team had about future directions to explore with this kind of research or potential clinical implications, was there any sense that there might be some applicability for more sort of purely considered neurodevelopmental disorders along the lines of, say, autistic spectrum disorder or those sorts of things?
Yeah, we've spoken to, there's obviously a lot of interest in using TMS for things like autism and ADHD as well, is a question that we get a lot. And speaking to clinicians, they say, but TMS alters dopamine and it targets these brain regions. And we know from the MRI studies, it activates these pathways. Why aren't you using it for ADHD? And I don't know the answer to that. I think again, it's this sort of clinical trial versus animal models and points that we haven't got the animal. There are animal models for things like autism and ADHD, but again, they don't always model all the different aspects of the human disorder. And treating people with autism and ADHD is, especially a lot of them tend to be young people, the ethical considerations around that are quite complex. So I think there are a lot of difficulties in moving into that space, and getting TMS approved for use with depression is probably a first step in moving towards that. It's the easy way in that population. But certainly clinicians that we've spoken to are really excited about possibility, and parents and children as well. The people need, I think, better treatments overall for those neurodevelopmental disorders. And TMS certainly has the capacity to modify the developing brain, as a lot of people have shown. We're not the only people who have done work in development. There's a lab in Germany that did some work in developing rodents, looking particularly at the critical period, and they showed some really interesting effects around the inhibitory excitatory balance, particularly parvalbumin into neurons. Alia Benali and Klaus Funke did some great work on characterizing changes in those interneurons that we know are involved in autism and regulating activity of brain pathways. So there's a little bit of preclinical evidence, but translating that is going to be complex. And you want to get it right, I think, particularly for something like ADHD, where it's all about balance, autism probably as well. And it's important to get it right.
Yeah. I'm curious in terms of follow-up studies or ideas based on the study that we're talking about with the visual cortex development. Would it be feasibly something to think about in terms of potential for a follow-up study that looks at perhaps different systems such as auditory processing or executive function, thinking about other aspects of brain function that are critical for, say, learning and success in academia or social settings?
That's a great question. I work with a colleague, an auditory physiologist, and we've done some work on tinnitus and TMS. And I've spoken to her about some way that we could use the auditory system. There are techniques like mismatch negativity and oddball paradigms where you can measure the brain's response to different auditory stimuli. So we've spoken about that a little bit. But you're right. It's the idea of looking at more cognitive and emotional type tasks around executive function and social interactions as well. We're starting to develop those paradigms in the lab. We're almost spoiled for choice. I can't say, we've gone in the direction of extending the time frame of the studies, as I said, for a couple of years. And we've focused in that long-term study on a particular model of anxiety, which is a prenatal stress model. So the animals that we're taking out to 2 years with TMS do actually have altered behavior, things like the elevated plus maze, where you measure anxiety, the amount of time that rodents spend in open spaces indicate anxiety. And we're also looking at social interaction in those animals. So how much time they spend with familiar or novel conspecifics. So animals, other mice that the mice have never met before. So we're trying to build in a wider range of behavioral tests that are more relevant to mood disorders. And again, I think once we have a better idea of what's happening over this 2-year period, we can then go back in again and focus on expanding particular areas or focusing on a particular time point as well. And the big question that everybody asks us is, how about maintenance, TMS maintenance? How are you going to give the mice maintenance TMS at 6 months? Oh no, you've just added another 60, 100 million mice to our experiment. It's just, there's only so much that we can do, and there's so many questions to answer.
Yeah, it must be a challenge to think about in some ways how to balance the focusing on the safety with interesting changes that might correlate with effectiveness in human clinical models.
And I think that the safety issue, we're really confident that TMS is safe. The changes in behavior that we've identified, we're hoping that we'll be able to show that they spontaneously revert. If not, I think the sorts of changes around integration in sensory motor brain function, we know that there are very good physiotherapy type interventions that can act on those things. So if we do need to combine TMS with some sort of rehabilitation therapy afterwards to make sure that those pathways are restored, I think that's very feasible because there's a good background on those sorts of interventions. So the safety, I think we're confident that there's not going to be any major problems. It's just making sure that the efficacy is really targeted. I think that's one of the big goals of the lab.
So would you say that there's some validity in the notion that then perhaps the less focused magnetic field might be almost more akin to TDCS, where there's perhaps more of a priming or potentiation, so that in concurrent kind of activity, as you say, with physiotherapy or behavioral therapy or some sort of active enrichment paradigm is going to be where there's kind of a synergistic effect for in like a positive therapeutic sense.
Yes, that's a really good question. And we've had the low intensity TMS compared to TDCS or TACS, the transcranial alternating current, in a number of conferences and discussions, reviews that we've had. So I think that's a really interesting point. And it comes back to how much do we actually need to stimulate the brain when we're delivering TMS and, activating motor evoked potentials and triggering action potentials. is that overkill? Are we actually activating the brain so much that you then call up homeostatic mechanisms that might try and reverse that plasticity because we've gone too far? So the low intensity stimulation from that point of view of priming and raising the capacity of the brain to respond in a more sensitive way to stimuli is a really tempting way to look at those mechanisms, I think. And homeostatic plasticity is very complicated. I don't think I really understand it still. And it would be wonderful to think of TMS as a primer. And we've done some work in animal models where we deliver TMS concurrently with a learning paradigm, concurrently with wheel running, so exercise. And we also have delivered TMS to the visual system in the dark. So the animals aren't getting concurrent light stimulation while they're getting their TMS. And I'm very proud of that work. I think it's really, really interesting work because by playing with the endogenous brain activity at the same time as you're delivering TMS, we can defy you can have too much activation, but you can also have too little. And it's sort of Goldilocks bit where you get the right amount of endogenous activity combined with the right amount of TMS in order to repair brain circuits in the best possible way.
Yeah, that's fascinating. And so much of what we know about the brain and how, that the tried and true treatments work, it does really reinforce this idea of just trying to restore homeostasis and balance. And so that makes total sense. Yeah. Just also trying to not interfere too much with what our body does naturally to try and maintain its own equilibrium. Definitely. So I'm just curious, like, as far as you've mentioned, a few times, understandably, certain kinds of limitations around things, funding and that. And so I guess just in an imaginary world, if you had unlimited funding and no barriers, what ambitious experiment do you think you'd be interested in doing? Or, what would be something that you maybe would want to try and pursue based on what the lab has found so far?
Yeah, that's a really good question. And I love those open-ended questions. Wow, what would I do? And I'm actually the 2-year study, the long-term lifespan study in mice that I've described a few times today. That was really my dream experiment that I would do. And I'm very fortunate we received a large grant to do that work. And it's an exceptional opportunity, I think, for us to answer that big question of lifespan. It's not a small undertaking. But even, bigger than that, it was interesting. I was at a conference at the end of last year where we had a TMS symposium with researchers from various labs in the US. They very kindly invited me to join in that symposium. And the impression I took away from that was that we're all doing very different experiments. So some people are using optogenetics to trigger activation of neurons in the brain to study particular frequencies. And other researchers have done similar to what we've done, miniaturized the coil, but managed to engineer it in such a way that they get a much higher intensity magnetic field. And they're looking at the processes involved in that. There are other groups that are using electrical stimulation, so bypassing the magnetic field altogether in animal models and activating neurons directly. And all of the talks were just so fascinating because they addressed a particular question in so much detail that nobody's solved any problems, but they're really increasing knowledge. And I think at the moment, what's so exciting about the TMS field is that everybody's doing different models and different approaches. And in a few years, I'm really hoping all of that will come together. And the big experiment that I would like to do almost is to regularly get these preclinical animal researchers together and make sure that we're doing things in a way that we can share that information across models. Because I think all of the models have value. If you're asking a question, there is a model that will answer that particular question in the best possible way. But we all have different questions. So we're all using different models. But, it would be wonderful to just put all of that together in a way that we could then make sense of the bigger picture based on those individual puzzle pieces. I think the TMS field at this point it's a bit wild. Everybody's doing their own thing, but it will come together. And I think maintaining that communication across labs and facilitating travel and exchange and collaboration will really move the field forward. I don't have a big vision yet. I think that vision will emerge, but I'd love to be part of bringing that vision together.
hmm Yeah, that makes perfect sense. And it makes me think that one of the things you said earlier about how the evolution of TMS has almost kind of gone backwards where, it was just sort of devised as the device, initially when 25 odd years ago, and then just trialed on people. And then the research has kind of gone backwards, right? And so now it's that kind of time course makes sense in terms of how then there's this sort of perhaps somewhat disorganized kind of scatter shot of approaches in terms of other technologies or variations on theme of TMS. But, hopefully, I think obviously, the more information that we have about these therapies, these technologies, then the better off we all are, because then we can just, the synthesis of it, I think, as you say, is really important in terms of separating the wheat from the chaff, I guess, and then figuring out how and to best apply that in clinical terms to get people well as quickly as possible for as long as possible.
Yep. I think also there's the temptation in TMS to dive into that parameter space where you can test infinite numbers of frequencies and pulses. And no, I don't think we have enough time to do that. Literally, I think it would take just a ridiculous amount of time. So every so often we sit down in the lab and discuss how could we design an experiment, or how could we use the tools like AI and modeling to work out what parameters are the most likely to be effective. And I think sort of more strategic thinking around what are the best experiments to do. It's important to do that from a translational point of view, because it's so easy to get lost in all of these very complicated parameters. And as you mentioned before, there are a number of stimulation modalities. You've got the TDCS and TACS. Ultrasound is becoming this latest fighting new tool. And are we back again to the device that's going to start being used in people without having the research to back it up? So I think also there's cautionary tales there moving forward. If we have got new technologies that could stimulate the brain, let's maybe just take a step back and do some of the basic safety and mechanisms so that we know what we're doing.
Yeah. No, well, congratulations to you and your team for that, because I think it's extremely important to just guard against the potential for there to be excesses and people to be, taken advantage of if it's someone that's maybe unscrupulous or maybe just overly excited and developing devices that are not necessarily regulated or perhaps as evidence-based as they should be. And then so having research like yours that is obviously solid and is just going to add to the body of work that shows that there is an evidence-based, not only kind of clinical underpinning based on population-based studies, but the basic science is also aligned in terms of the evidence, not only in terms of the effectiveness, but the safety especially.
Thank you. That means a lot to me, and I'm sure my team will appreciate that as well. The other thing I want to mention is that we publish a lot of negative results, and that I think is also really important. It's always a bit of a struggle to get those results published, and the students often who lead those studies often feel a little bit disappointed, but it's really important to point out when things don't work or don't have an effect. And I think that is something that, again, we're really proud of publishing as much negative results as we can when possible.
Yeah, that's a really important point. Maybe for viewers and listeners, can you just explain a little bit about what that means, negative studies, and a couple of examples from your lab?
Yes. So I have plenty of examples. I suppose the recent study, the adolescent mouse TMS study, really, initially, we thought that was going to be a completely negative results study. So when we stimulated the mice and didn't see any changes in the anatomy, didn't see any changes in the cellular components, but only a very small behavioral change. Now, that was essentially, most of that study was negative. And what that tells us is that TMS is probably not having massive effects on certain things, which in our context was a good result in safety. We've had other studies, for example, where we've given stimulation during I'm thinking of the task. It's a lever pressing task where you train mice to press a lever to get a reward. And we thought that because generally TMS is thought to increase dopamine levels and might affect the reward pathway, we thought this is a great model to look at so that we might be able to see that delivering TMS during lever pressure might reinforce those compulsive or addictive behaviors, or it might prevent them. And that was a study that was negative. So that allowed us to say, well, in our models with our TMS, we're not going to waste any more time looking at addiction or looking at compulsive behaviors because our particular tools don't work. It doesn't mean that human TMS doesn't work, but in our models, we cannot use that to study addiction. So those sorts of examples, I think, are really useful.
Yeah, definitely. No, again, congratulations. I think it's just really impressive. And, it's very exciting just in terms of, I'm just sort of dipping my toe into understanding more and more about the basic research, the animal models, and it's just endless in terms of potential, and it's extremely interesting. Well, maybe before we wrap up, was there anything else that you wanted to share about, your work, your lab's work, and anything that you thought that might be important?
No, thank you very much. I just like to acknowledge that, I've spoken a lot about myself and as if it's my work, but obviously it's the work of many, many students, very talented honors and PhD and master's students and postdocs in my lab over the years. And also in research, people bring their own interests to the team. And it's really been through students asking particular questions and their own interests that we've gone off on little side tangents and found some really interesting findings. So being in science is really exciting, and the opportunity to do these experiments and ask these questions is a real privilege. Thank you for letting me talk about it.
Oh, that's great. Thank you so much for talking to us today. I really appreciate it. And again, congratulations to you and your colleagues, students, everyone. I think it's just fantastic projects that you've spearheaded. And I think that hopefully we'll find out more in the coming months and years about the upcoming results, especially of the 2-year study. I think it's going to be very interesting to see how that goes. So I'm definitely keep myself posted on that.
Thank you. I'm excited as well.
Great. Okay. Well, thanks again, Jenny. Really, really appreciate your time and such an interesting conversation. So all the best. And hopefully maybe we'll bump into you at some point at a conference or have another podcast at some point in the future. Maybe we can plan to talk a bit about the results of the long-term study when those are available.
Love to. That would be fantastic, Mike. Thank you.
Okay. Yeah. Thanks. So that brings us to the end of today's conversation with Dr. Jenny Rodger. Her research offers crucial insights into how low-intensity rTMS interacts with the developing brain. And it really reminds us of the importance of cautious, evidence-based innovation as we expand neurostimulation into younger populations. If you've enjoyed this episode, please be sure to like and subscribe, leave a review, or ask questions in the comment section below, and share it with anyone that you think might be interested. I'm going to add links to Dr. Rodger's lab's information and contacts and the relevant papers. I'll leave links to all of that in the show notes below. So please do check those out if you're interested in diving deeper into this really important subject. Thanks again for your time, your interest, and your attention. I really appreciate the fact that you watched or listened to the podcast, and we'll see you next time on the Neurostimulation podcast. Thanks so much.